So let's get back to it. So last time we finished up talking about the plasma membrane, transporting things across. Now we're getting into the inside of the cell. So remember that inside of the cell, we have the cytoplasm, the intracellular fluid, and this is where we're going to have all of our organelles, right? Our mini organs that handle the jobs that are required in order to keep the cell alive. So these are mini organs, and so this is going to be a key component as we're going through knowing all of the different organelles and being able to describe what they do. This is very similar to how when we start doing organ systems and we're saying, okay, this is the digestive system, there's the stomach, what does it do? There's the pancreas, what does it do? There's the jejunum, what does it do? There's the gallbladder, right? You need to be able to know what they do. Not only the actual names, but be able to describe what they do using anatomical terminology if necessary, right? Okay, so we have organelles. We also have, in some cases, there are inclusions in the cell, and then the cytosol itself. So the cytosol itself is the gel-like material where all of these different things are sitting within. So when we look at these mini organs, that's where we're really going to focus our time. We have it categorized into two ways. There are those that are membranous, that have their own membranes to them. So we're going to see things often go into them and then come out of them. And then we have the non-membranous organelles as well. Non-membranous include ribosomes, the cytoskeleton, and the centrioles. So you can see most of what you've seen in the past in terms of organelles are called membranous. And what we have is, with these membranes, it allows each organ to do something on its own and not contaminate the rest, or it's able to perform its particular function in itself. And so that's the same thing like with the heart, with the blood in it, with the stomach, with the food in it, whatever. Okay, so this should be fairly familiar to you from your high school biology, so I'm going to move through it fairly quickly. But again, if you see something that you're not familiar with, make sure you add in those new levels of detail that we're going through, because we're going to be talking about these organelles in particular specialized cells later on. So for instance, the mitochondria, power plant of the cell, because they are producing most of the cell's ATP, right? The adenosine triphosphate molecule, that energy storage molecule. Now when we say most, that's because when we talk about cellular respiration, there is anaerobic production of ATP as well that occurs out in the cytosol, in that gel. All right, so when we look at the mitochondria, we know that the mitochondria, they're a membranous organelle, but they actually have a double membrane. So you can see they have an outer membrane and an inner membrane. And the inner membrane has all these folds to it that are called cristae. And so there is transport of substances like ions across these different membranes in order to help produce ATP. Lots of enzymes that are embedded within it as well. So the enzymes, remember enzyme, what type of molecule is that? And what does that do? So that's a protein, remember? And so it's a protein that works to decrease the energy necessary for a chemical reaction to occur. So in any case, these are very specialized bean-shaped organelles that produce ATP. And when we look at them, it's interesting. Scientists think that once upon a time, mitochondria, because they have their own DNA, right? So they have their own DNA, and in fact, they have their own RNA and ribosomes. When you look at them, you're like, wow, that seems much like a cell itself. And they think that once upon a time in evolutionary history, mitochondria were a unicellular organism that possibly got ingested by a larger cell and then incorporated into it rather than digested, and end up setting up a symbiotic relationship with that larger cell. And then that continued through time. So that's why it has a double membrane. So for instance, they think that the inner membrane that we see here was this unicellular organism's original membrane, and then this is the membrane that the larger organism, when it's, the vesicle, etc., when it encapsulated it, that's where that second membrane came from. That's the idea. And then mitochondrial DNA, right? Mitochondrial DNA you inherit from your mother instead. So that is maternal DNA that we see inside of there. So there's lots of interesting things about mitochondrial DNA and the mitochondria themselves. like we said, in a lot of ways, they almost operate as their own little organism inside of our cells. Okay, but aerobic production of ATP is their job. Then we have ribosomes. So, like we said, there's ribosomes inside of the mitochondria, but they're also floating around in the cytosol, and they're also attached to other organelles, particularly the endoplasmic reticulum, also known as the ER. So you can abbreviate it ER, but you should know that ER means endoplasmic reticulum, not emergency room, etc. So the free ones, like we said, are going to be floating around in the plasma, if you will, the cytosol, the plasma inside the plasma membrane, if you want to consider it that way. And the membrane-bound ones are attached to particular organelles like the endoplasmic reticulum, making it look rough. So that leads us to the endoplasmic reticulum. Again, membranes, and these are membranes that are associated with the nucleus, attached to the nucleus. So it's continuous with the outer nuclear membrane. And so there are two varieties, rough ER and smooth ER, and like we said, the rough ER is due to attached ribosomes. Smooth ER then is not going to have ribosomes. Now the main role of the ribosomes are to produce proteins. And so when we have rough ER is the main production area inside of the cell for producing proteins. Proteins for the cell, proteins for export, etc. Like enzymes, hormones, all kinds of things. So we can see it here when we look in the microscope. It actually looks rough because you can see all these little studded ribosomes along the membranous surface of the ER. So rough endoplasmic reticulum, and then smooth endoplasmic reticulum as well. So rough endoplasmic reticulum, like we see there, is going to be the site of protein synthesis, both for, like we said, use of in the cell, and then also to be secreted from the cell. So they are produced in those ribosomes attached to the endoplasmic reticulum, and then if they are going to be further processed, like a lot of times for export and things like that, they're passed from the rough endoplasmic reticulum to the Golgi apparatus, another membranous organelle. Now the smooth endoplasmic reticulum, like we said, do not have attached ribosomes, and their job is quite a bit different, right? So rather than proteins, we see it associated with lipid, fat, right? Lipid metabolism. So that if we're talking about some hormones are made of proteins, and some hormones are made up from a basis of cholesterol. It starts with cholesterol, and then we attach functional groups to it, and it becomes like steroid hormones like testosterone and estrogen, etc. So that comes from the smooth endoplasmic reticulum. Also, we can break down fats there, detoxify certain types of chemicals, work on converting some of our energy molecules, and storage and release of calcium in particular cells like cardiac muscle cells and skeletal cells. But this one here that we see here is the main one, lipid metabolism that we see here, as well as the processing of lipids. Those are some of the critical ones. So you can imagine smooth ER, there's going to be a lot in the liver, you would imagine, and associated with the digestive system. Okay, Golgi apparatus. We said Golgi apparatus is further processing of those proteins. So in a way, you know, what we'll find is that there's a number of different organelles that are associated with protein production. Proteins are critical for the activity of the cell itself, plus for use in the body overall. And so that's why you'll see so many different organelles are in some way related to protein production. So it's like a whole factory. And in fact, the nucleus, remember the rough ER is attached to the nucleus? The nucleus is basically where we have the blueprints. That's the headquarters for the protein factories, right? So that's where we have the recipes that says what proteins this particular cell is going to make. Because to a certain extent, that's what DNA is. DNA is, number one, a recipe on how to make a new cell, and number two, a recipe book of how to make all these different types of proteins. And those individual recipes are genes. So if we're going to produce a particular protein, we're going to read that gene, and then we're actually going to produce those proteins, right? Here's the recipe. We're going to send it out to the rough ER, which is the protein factory, or even the free-floating ribosomes, and then those are going to crank out that particular protein by attaching amino acids together. And then the Golgi apparatus, once we've produced our product, like at a factory, we have to package it, right? So we're going to package it We're going to finish modifying it, right? We're going to dust it off and shine it up and stuff like that and attach groups to it. And then you're going to wrap it up into vesicles and move those vesicles out for export from the cell in a lot of times. And so the Golgi apparatus is kind of doing that as well. It can do that for proteins. It can do it a bit for lipids as well. But it's a processing plant, the Golgi apparatus is. And so, you know, this is like, okay, we've already done the whole production line of these products. Now we need to package them. We need to put them in their individual boxes. We need to put them in big shipping containers. And we need to put them on the truck to get them out. So that's the role of the Golgi apparatus itself. So 3 steps involved there. We have these little vesicles that bring the produced proteins from the ER to the Golgi apparatus. Then we do that kind of like shining up and sorting them and packaging them, etc. And then we basically move them out to be to get them to the plasma membrane. And that's what we can see here. So you can see how it all starts. There's our nucleus. There's our rough ER. We're going to transport it from the rough ER using these transport vesicles. We're going to get it to the Golgi apparatus. We're going to process through these flattened sacs that we see here. And then we're going to have secretory vessels that are going to go up to the plasma membrane and release those proteins, etc. So you can see that whole relationship there of these different organelles, but they're all associated with protein production. You can see some of them can actually go inside of the cell too, right? Okay, other types of membrane-bound organelles, we have peroxisomes. Peroxisomes are kind of like, if you will, kind of like the immune system, if you will, of the cell. So they will help to get rid of particularly toxic substances that can be inside of the cell. A lot of which are what are called free radicals. So you hear about free radicals when you're talking about different types of superfoods, etc. These are ones that can really damage cells, can potentially cause them to become cancerous maybe, but the peroxisomes work to actually get rid of those free radicals or at least detoxify them. So there are different enzymes in there, including oxidase and catalase, and that's one thing to note, right? Anytime you see a molecule and it ends in ASE, that is a suffix which tells you that it's an enzyme, right? So particular proteins in there that will work to detoxify. Detoxify to, they use actually hydrogen peroxide, and then hydrogen peroxide, we got to get rid of that. So there's a second enzyme which breaks that down into water. So we get rid of one of those oxygens that we see there. They also can work on breaking down and synthesizing fatty acids, which are associated with fats themselves. But a little bit like the immune system of the cell. And in addition to that, we have lysosomes. So we had peroxisomes, now we have lysosomes. So these ones are a little bit more like the digestive system. So the digestive system of the cell, like the stomach, are going to have enzymes associated with them. Again, ASE, like we see here, that are going to be able to break down things. So if we brought in, for instance, different types of chemicals for metabolism, we can break those down and release them. It can also work as a cleanup crew to get rid of old organelles that aren't working. And it can get rid of, if we're talking about like white blood cells, it can get rid of pathogens like bacteria and viruses and toxins. So what will happen is these lysosomes, these little bags with these enzymes within them, these hydrolases, what they're going to do is they will, if it's a white blood cell, it'll come and it'll latch on to a bacterium or a virus and bring it inside, and then that lysosome will come to it and will put that pathogen in the bag of enzymes in the lysosome, and it'll digest it. Just like the stomach, you can imagine the stomach acid digesting things. And in fact, like we said, the enzymes there are also acidic, acid hydrolases. Okay. So what we've seen then is that these membranous organs, a lot of them are associated with proteins, right? So we had the ER, both rough and smooth, the Golgi apparatus, secretory vesicles, those are also little bags, right, that are membranes, and lysosomes. So these ones that we see here, as well as the peroxisomes, they are sealed off. They have an inside because they have a membrane that surrounds them. And that way they can do their work without disturbing the rest of the cell. So these are some of the membrane-bound organelles that we've talked about so far. But now we need to move into all these other things that we see here out in the cytosol. So we move on to the cytoskeleton. So again, we're talking about these living cells that have many organs that do these different jobs, like immune system and digestive system. And there's a brain, if you will, kind of like the nucleus. But much like our bodies, in another way, they have to have some sort of structure to it that aids in the movement of the cell or the change of the cell shape, etc. So they have a cytoskeleton. So again, when we look at the words, when we look at that word, when we see cyto up there, that means cell, right? So the cytoskeleton is not going to be made up of bone, but it's going to be made up of proteins. More proteins, right? So you can imagine, just like our own skeleton has to be remodeled and strengthened and stuff like that, happens with the cell as well. Those proteins that give it shape and that help it to move wear out through time. So even more reason why we need to be producing proteins with all these other membranous organelles. Okay, so rather than bones, they're more like a series of rods, etc., that help to, again, give structure to the cell as well as to help it move. And there's three different types: microfilaments, intermediate filaments, and microtubules. So they're kind of three different sizes, three different shapes, all have to do with supporting the cell in some way. So first off, we have the microfilaments. These ones are some of the smallest ones, and these ones will either change the shape of the cell, or it can also help the cell to move itself. So there are obviously the muscle cells of the body that are going to use a lot of these microfilaments, but there are also things like white blood cells that will actually extend out and do this amoeboid movement as they pursue pathogens, and they'll use microfilaments as well. And you can see how they're pretty small. So when we see NM there, that's nanometers, like 1/1,000,000th of a meter. And so they're pretty small, and these are all individual proteins that are kind of threaded together. So microfilaments that help with movement and cell shape. Then we have the intermediate filaments. These are going to be a little bit bigger. They're more like ropes. And so rather than just like two like we saw, it's going to be more proteins that are coiled around. And in this way, they help to give some strength often to the cell itself. So you can see here some of the additional structure associated with it and see how they're a little bit bigger. And here you can see, for instance, some of the intermediate filaments that are forming, helping to form the structure inside of a particular cell of the body to give it a particular shape, to give it some strength, etc. So inside of neurons, we have these things that are called neurofilaments that give it its structure. So they're supposed to be these long cells with all these star-like extensions so they can communicate. And then epithelial cells, keratin cells, particularly those that are along the edge of your body, need to be tougher as well. So they'll have a bunch more intermediate filaments in them that are called keratin filaments. Again, strengthening. And then we have microtubules. Now this one, right, we talked about filaments before, which look solid. These are tubules, small tubes instead. So these are the largest ones. And these ones help to kind of organize and distribute things inside of the cell. So if we look at it, see how it kind of coils around instead? So a little bit bigger, right? 25 nanometers, so it's 7 to 10 to 25. And these ones, like if we're talking about dividing the cells or moving things around the cells, they help to form these like highways. They help to organize the chromosomes when we're dividing the cell during mitosis and things like that. Okay, other types of proteins associated with the cytoskeleton. These ones are cool. Motor proteins. So motor proteins, right? If you see over on the right-hand side here, there's a lot of cool little animations that they have online, right? How cool is that thing? Just kind of going along. But you can see how we see some of our other ones. Here's a microtubule providing a pathway for the transport of a particular vesicle, right? We got to be able to get it. We don't want those vesicles bouncing around inside the cell. We want them to go from one place to another, from the rough ER over to the Golgi apparatus, from the Golgi apparatus and the secretory one to the plasma membrane. And so these motor proteins help to move it around, and they're powered by ATP. It's always the coolest animations that I see there. Okay, some other parts of the cytoskeleton that are important that we'll talk about coming up are the centrosome and the centrioles. These ones are part of what we call the cell center, and these ones help to, like we said, organize like the different organelles, organize the chromosomes when the cell is dividing, as well as they're the basis for some of our extensions of the cell, which are called cilia and flagella, that help cells to move. So cilia and flagella, they're very similar to each other. The structure is very similar. It's kind of like if you look at tires, sometimes they'll show you all the steel belted radials and stuff like that. But look at that interesting organization that we see here, right? So you can see microtubules inside of them and organized in a circle like that or in triplets like this. So in any case, these, you can see, are complex structures, proteins, that either will form individual flagella, right, like on a sperm cell, or they will form cilia if we're talking about shorter ones that are more numerous. And because they are proteins, like we said, we can actually move them with the help of ATP, and as we move them, they can move things often past the cell. If you're looking at pond water or something like that, sometimes you'll have ciliates, ciliated organisms, unicellular organisms that have cilia that move the whole cell, and they'll kind of float around. But in our cells, most of them are attached. So the cilia that are moving things past the cell. So for instance, like in the trachea, we have cilia along those epithelial cells lining it, and they act as a mucus escalator. So those cilia are constantly moving mucus that's secreted up away from the delicate respiratory membranes and up and kind of out. Catches particles and things like that. So cilia and flagella about movement. We also have microvilli. Now microvilli will also have proteins inside of it, microfilaments inside of it, but these are not for moving, they're more for increasing surface area. So we see them a lot in the digestive system, the urinary system. And so if we look at the surface area of this cell, and we look at this side along the edge, right, you can see that the surface area, you're only going to get that much area. But if we look at this apical surface, look, it goes up and down and 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 up and down. If we stretch that line out, it would be like this long compared to what we saw here, which was maybe just this long. So we increase the surface area. It's a lot more places where we can pull things across into the cell, etc. So those are the microvilli. All right. So now what we've done is we've gone through and we have taken a tour of all of those little mini organs inside of the cell. So remember, you need to know the name of them, you need to know what they do. The last bit of this lecture gets into the nucleus and its role in terms of, like we said, bossing the cell around, right? It's a cookbook on how to make new cells as well as how to make these proteins. So we need to remind ourselves about DNA and things like that, how we copy DNA, remind ourselves about mitosis too. So we'll see that coming up in the next segment.